Okay, so maybe we can begin. So firstly, thank you very much for registering and joining our first webinar in the series, which we're very pleased to be co-hosting with Frontiers Computational Neuroscience and Frontiers Neuroinformatics. We're delighted to see that so many of you have joined. So my name is James Isbister. I'm a scientist at the Open Brain Institute, and I'll be giving a brief introduction to the Institute and our virtual labs. Then Darshan Mandge, my colleague and scientist, will be giving us a very exciting scientific demonstration in his virtual lab. Maybe he can say hello. Hello. Then we'll have a short presentation from Olivier Boylan about the journal Frontiers in Computational Neuroscience. Maybe he can say hello.
Hi, everyone.
Thank you. And then Sara will be presenting Frontiers in Neuroinformatics. She could say hello.
Hi, everyone. Nice to meet you all.
Thank you. So if you have any questions, please write them in the chat and we'll have 10 minutes to answer them at the end. Please be aware that we'll be recording both the presentation and questions. And at the very end, we'll have a brief poll with 5 questions in teams, which would be very helpful to get feedback on the webinar. So in this webinar series, we'd like to convince you that the era of simulations neuroscience has arrived. And we'd like to make 3 main arguments. Firstly, that general multi-scale models are now well proven as a powerful scientific tool. And I'll explain what I mean by multi-scale models. That new types of data can improve the efficacy of such models by orders of magnitude. And that with the right collaborative methods, we can speed up the development and exploitation of such models by orders of magnitude. And that is our focus at the Open Brain Institute. How we can enable the scientific community to collaborate in order to leverage existing models, build the next generation of models, create impact for users with their results and models being instantly reusable by the community, and for people who are new to this type of modeling to be a place for learning and teaching. So who are we? We're a team of 35 scientists, engineers, and designers based in Lausanne, Switzerland. The Institute was founded by Henry and Camilla Markram in January 2025. Henry founded the EPFL Blue Brain project, a Swiss-funded project, which completed in December 2024 and contributed to the field of simulations neuroscience for 20 years. The Open Brain Institute continues the work of the Blue Brain project and is led scientifically by our Chief Scientific Officer, Dr. Michael Ryman. And we should offer the disclaimer that whilst our code and data are openly available, we're a non-for-profit which offers a paid service, which importantly is totally free to try. So after 1 year, we have a host of exciting customers and collaborators with users who have set up virtual labs all around the world. And we have already 560 virtual labs created. So what do I mean by general multi-scale models? Well, I'm referring to networks of multi-compartment neural models. And this simply means that each neuron is modeled as an electrical circuit, similar to what you will have seen in your physics classes at school. And Darshan is going to go into more detail. But essentially, every point in a neuron has a voltage, and current can flow through the neuron or across the membrane through ion channels and synapses. Researchers typically optimize the density of different ion channels in different parts of the neuron in order to recreate neuronal responses observed in single neurons in vitro under particular protocols. Anywhere from 2 to several million of these neuron models can then be placed and connected in a 3D space following the true geometry of one or multiple brain regions, such that we have a simulatable model which spatially has a one-to-one correspondence with a real brain region, in this case, the somatosensory cortex of the rat. So why then do we call these models general? Well, they're general first in the type of questions which they can be used to explore. They can be used to integrate and relate different types of data, such as neuron shape, connectivity, and electrophysiology, infer missing data based on patterns in the available data, constrain brain theories, provide explanations of observed experimental phenomena, act as a ground truth, which I'll describe, identify knowledge gaps, and perhaps most importantly for any model, make predictions. And these models are prediction generation machines. You can change any parameter from the level of ion channels to morphological shape or connectivity and measure the effect on any signal from dendritic dynamics to extracellular activity or EEG. And any prediction has an exact location in real brain tissue where it can be tested biologically. And because of this multi-scale nature, they're also general in terms of the topic of research. So the single somatosensory cortex model, for example, under a single well-characterized parameterization, has been used for the following types of study, which there are links to through the QR code. The important point, though, is that these models allow exploration of research questions which are very challenging, if not impossible, to study biologically. So I'll give a couple of quick examples. So firstly, a key goal of neuroscience is to understand the dynamics of large populations of neurons at high temporal resolution. And many of you will know that the only way to do this is to use arrays of electrodes inserted into the extracellular space surrounding neurons, or the most common way to do this. And each electrode on the array then records the voltage at its position in extracellular space. Spike sorting algorithms are then needed to infer the presence of possible neurons around the electrode, detect spikes, and assign them to these hypothesized neurons. But are these algorithms good enough to give us an accurate view of neural activity? Well, in our model, we can insert an extracellular neuropixels array and record extracellular activity during a simulation. We can then apply different spike sorting algorithms to this data and compare the accuracy relative to the true spiking activity of the simulation. We can also ask how our view of the network's ability to encode information is degraded by looking through this lens of extracellular recording. Another key goal of neuroscience is to understand how plasticity at synapses leads to learning within the brain. Well, a lot of studies of synaptic plasticity have taken place in vitro with very different conditions to in vivo. By building a biophysical model of synaptic plasticity, we predicted that pairwise plasticity rules change dramatically under in vivo conditions. And by placing the plasticity model into our cortical network, we were able to make predictions about how plasticity at a synapse depends on its location in the overall network under in vivo conditions. So how then can new types of data improve these models? Well, from large-scale electron microscopy datasets, we can now reconstruct volumes containing hundreds of thousands of neurons. Using tools available on the platform, users can automatically extract precise morphological shapes of individual neurons, as well as all of the synaptic spines. So this type of data is a game changer for building these multi-scale models. We have the exact neuronal morphologies, their placement and connectivity to each other. We can constrain synaptic physiology based on synaptic shape. We can better compensate for neurons outside of the volume, as we can see all the synapses from such neurons. We can validate the emergent activity against functional activity recorded from the exact volume of tissue prior to its reconstruction. And we can use transcriptomic data to constrain the types of ion channels we place in different neuron types. So by now, you're hopefully asking, can I take part in this? Can I run my first brain simulation? Can I leverage existing models? Can I take part in building the next generation of models? Well, if you want to do it on your own, you might need the right hardware, code, data, models, the right versions of all the above which work together correctly, expertise perhaps to help you when they don't work correctly and to help you validate when they do, and collaborators to create impact together. Your experts or collaborators might be on the other side of the world. So now, how do you keep software, data, and models which are evolving on a weekly basis in sync? And how do you do that efficiently? So for large complex data projects, collaborations might stay limited between one or several groups because of these challenges. And if you get through all of that, how can you make all of this as simple and straightforward for other people around the world to use? So our proposed solution is called virtual labs, which I will first briefly introduce before Darshan gives us a live demonstration. So you don't have to do this now to follow the webinar, but when later you can navigate to our webpage and click the big button, Go to Virtual Labs. When you click this, you'll be taken to a page which prompts you to sign up for free with either your Gmail, GitHub, or Microsoft account. When you click Create Virtual Lab here, this will send a verification code to your email address. So please let us know if you don't receive the code, and you can contact us here. But once you're signed up and that's complete, you will arrive here in your new virtual lab. And particularly, you're inside your first project within your lab. And in this top left corner, you have a virtual lab and project selector. So if you click that, you can switch between projects within your virtual lab. So here I have 6 projects within my virtual lab, but I can also see the virtual labs and projects which I've been invited to. So, for example, I'm a member of Michael and Aima's lab here. But within this particular project, I have a set of members who I've invited, and I can also add more members to the project here. So what can we do in this project? Well, the first thing we can do is browse and create data. So it might not sound incredibly sexy, but for me, one of the most exciting features is that the platform is built upon a cloud database, which is accessible from anywhere in the world. And data is split between public and project data. Public data is curated by us, and project data is private to your collaboration. But not only can you read data like other remote databases for science and neuroscience, but you can write data, not just when you publish your work, but on a second-by-second basis as you collaborate. But firstly, let's talk about the existing publicly available data. So the database is already filled with a lot of published data. We have experimental data from neuronal morphologies to single neuron electrophysiology to electron microscopy data. And a major advantage is standardization. So for a given, what we call entity type, like a morphology, all the files and metadata are accurately curated so that any code that works with one entity of a given type also works with another. And this is maybe most exciting for our circuits or general multi-scale models, as I referred to them earlier. So if we click model here and then circuit, we see a list of various circuits available on the platform. We already have a 210,000 neuron volume from the NBS1 volume, which I spoke about earlier, as well as various smaller subcircuits which have been extracted from it. We have a blue brain model from the Blue Brain project of the RCA1 region of the hippocampus, and very soon we'll have the Allen Institute's primary visual cortex model. We also have anatomical models of part of the micron's EM volume, electron microscopy volume, and human cortical electron microscopy volume. Anatomical models, I should stress. So at the moment, these are not simulatable. And as they are standardized, these advanced models can now be easily compared and used with the same tools directly from your web browser. So how can this data be used? Well, the first way is through our standardized workflows. So this is the workflows page. You'll notice that there is a table for browsing the previous workflows you've created, so you can navigate quickly to them from here. You can also duplicate a previous workflow to quickly iterate on a previous workflow. But most importantly, when you first arrive, you choose a category of workflow, such as simulate, and then a type. So I'm going to choose single neuron, beta in this case. Then you select the input data for your task, in this case, a single neuron. You can browse the public and project data, choose one, and then use that model in the workflow. And then you arrive in our standardized workflow view. And this is the one for designing a simulation of a single neuron. But because the user interface is standardized, if you learn what to do here, you learn how to perform any of our workflows, as you'll see with Darshan. But I'll just highlight a single feature of this general user interface that is useful to know about. So any parameter can be scanned over. So if you hit this plus button in one of the fields, you'll be able to fill out multiple values, as I've done here. So this is designed to maximize flexibility. Whatever the workflow, you can scan over any number of different variables. So when everything is done, you'll have all green ticks, and you can hit generate. And this will take you to the next page where you'll see the different coordinates in the scan, so the different simulations in the parameter scan in this case. And you'll see later that you can select a subset of them and launch them, or launch them all at once, and then view the results directly here. And you can quickly iterate by going back to the configurations page, as Darshan will show us. But if you want, remember, we're in the context of your project, so all of the data is written to your project and everyone in your project can view it. If you want to analyze the data further, you can copy the reference to the campaign in the cloud database and head to the Notebooks tab, where you'll find interesting notebooks for interacting with this data. So that goes into your project data. You can also upload data by hitting this green button here. And of course, there is an AI assistant which can answer questions about the literature, about our data, and it can also design an experiment for you. So here I'm asking it to add a stimulus, and it makes a proposal in the user interface. If you have any feedback, you can hit this button here, suggestions, issues, challenges, and we can help you. That will bring up this page for that. And finally, you can contact us directly if you'd like to see particular features added to the platform, if you'd like to contribute features or workflows in Python in our open source repositories. You might be, we're also interested in other neuroscience workflows beyond this type of modeling, and you can access our data programmatically. And the technical details are pretty simple. We have the concept of an entity type in the database. So these are the different types and workflows which consume these entities and create new ones. So entity types are defined in our GitHub repository entity core. You can read and write to this database using entity SDK. And if you're interested in contributing new workflows, you can take a look at OB1, which I'm involved with. So with that, I'll hand over to Darshan.
Yeah. Hello, everyone. My name is Darshan Mandge. I am a scientist at the Open Brain Institute in Lausanne, Switzerland. For this part of the webinar, you can just follow the slides and the demo. And after the demo, we'll provide you access to the project in which I am going to work. And you can try the simulation there at your own pace. If you have any questions, you can post them in the Q&A tab. We'll answer them at the end of the presentation. We'll also be recording the video, which will mail it to you later. So let's begin. So in this webinar, we'll learn how we can leverage the Open Brain Institute's virtual labs to explore, build, and simulate models that are grounded in experimental data to test your hypotheses, to simulate pathological conditions at subcellular, cellular, and circuit level. We will mainly focus on the R581Q mutations in the KCNQ2 gene, which this mutation has been linked to severe conditions such as benign familial neonatal seizures and developmental epileptic encephalopathy. So this particular gene in human neurons has been known to encode the Kv7.2 ion channels, which then make the M currents. Now, one of the studies has shown that the human iPSC-derived glutamatergic neurons with this mutation show a reduced KCNQ2 currents, that is Kv7.2 currents, and elevated calcium-activated potassium currents, which are SKCA and BKCA type currents. But what are these ion channels? So ion channels are some pore-forming proteins which are on the cell membrane, which allow the movement of ions across the membrane, and then it generates currents which can be then inward if the ions are moving inward. It also depends on their polarity and outward currents. These can be activated by different stimuli, for example, voltage, intracellular calcium, temperature, mechanical stress, ligands, etc. On this part, you can see the response of ion channel to different voltage clamp levels. So this is a Kv7.2 current recording. And you can see for different voltage clamps, you see different amplitudes of currents. And we have different types of channels for different ions, such as Kv1 to 12.3, and similarly sodium, calcium, and HCN channels. There is another category of channels which are calcium-activated potassium channels. Remember, I mentioned before that there are intracellular calcium concentration activated channels. So these belong to those categories. Some are large conductance channels, some are small conductance channels. Now let's go back to the experimental observations. So it was observed that when this mutation is present in a CHO cell line, which expresses the KCNQ2 gene, you get a reduced current when you simulate voltage clamp stimuli. So this is the control condition, and this is the mutation condition. And when you have this mutation in iPSC-derived glutamatergic, which are pyramidal neurons, they show different behaviors in the action potential. For example, there is change in AP threshold, there is change in the width of AP, and you can also see some changes in the this part of the AP, which is called the after hyperpolarization. This one is FAHP, which is the fast AHP, and you also have some slow AHPs, which are very slow and occur after 1 second. Now, how can we test this hypothesis? We want to see how these changes can be simulated on our platform. So we will have a workflow. I'll just go to this slide, and this is what we propose. So we start by looking into some models on the platform, neuron models, which has those Kv7.2 channels and SKCE channels. And then we simulate these two conditions. One would be control and one would be mutations with the changes in the conductances of these two channels. And once you have those simulation campaigns done, we will analyze those results in Jupyter notebooks. So let's start. So this is the home page of your project. So I'm currently in the OBI virtual lab, and this is the project. So the project title is Webinar 1, 2026, Ion Channel Mutation to Neuronal Function. And you saw that we want to first find some models which contain those ion channels. So we go to data. We have these brain region selectors here. We leave it as it is. Since we want to find some models, we go to model and we go to E-model. So E-models are the electrical models. And here you can see that there are different brain region single neuron electrical models. I know that for pyramidal neuron, I have a filtering I can do, which is based on the electrical type, which is called CADPYR. So this is the this is the E-type that we call it. So this is the continuous adapting pyramidal cell electrical type. And once you do that, you will see that there are several models on the platform which you can see. Now, our goal is to find a model which contains those two channels. So let's see. Let's randomly select this one. And you can see here, once you go to view details, you will be shown the page for the E-model, which shows what is the description, the brain region, some metadata, and what is the M-type, E-type. And then you can go to configuration, which will show you what is this E-model made up of. So you have this detailed morphology, you have these traces. These are the electrophysiological traces based on which it was made, and you will see ion channel models. So our goal is to find whether this neuron or this E-model has the Kv7.2 current, but I don't really see that here. So I see that there are only sodium channels, and there is this SKE2, which is the calcium activated potassium channel, small conductance channel, but we don't have Kv7.2. So and I know that based on my previous search that there are no models on the platform which have these two channels together. So what we can do is we can change our workflow, and what we will start by first. Let me just go to this slide. So we have to first build an ion channel model, and then we will add that channel Kv7.2 channel to the electrical model. And then once that is done, we'll make the neuron simulatable. And once that is done, we will simulate these two conditions, and everything stays the same. So let's go back to our platform, and we will start building the ion channel. We go to workflows, and even we go to build and we go to ion channel. Before that, let me quickly tell you about the ion channel properties here. So we have a slide which explains how the ion channels can be modeled. You can model an ion channel using Hodgkin-Huxley formalism. So this is a well-known formalism for modeling ion channels, where you can model the channels based on their activation and inactivation properties. So as you saw it before, you have voltage clamp stimuli, and you have the responses of the channels. And then you can have the channel parameters, activation and inactivation type constants, and the steady state parameters. And once you have the equations set up, you can then build the model. So now let's go back here. We have here under workflow, build ion channel. We'll click on that, and we will give a name here. Let's give it KV 7.2. Just a name, and I'll say a name here, webinar KV 7.2 campaign. Now I have to select a recording because we want to build the model from scratch. We have to first find the electrophysiological data which we can use to build it. Fortunately, the platform has several recordings for different ion channel types. And since we want to find our particular ion channel, which is KV 7.2, we'll filter it by that. We will also filter by the temperature, which is 35, so that we only find that particular channel. So once it's done, you will see you have these recordings filtered by that. And I will select one which looks very similar to the experimental recording that you saw. So we select that, and you can see here the recordings appear here. So these are the responses for voltage clamp, and you can see different amplitudes for different voltage clamp levels. So this ion channel recording has been selected. Now you have to define what equations to use. So we have options to add this equation. Since there is only one, we'll select that. This is a sigmoidal curve that we saw earlier in the slide. We select that. We have an equation to model the time constants, which is we have 4 options. We'll select the first one here, and we have equations to select for the inactivation and time constant of inactivation. So we select that, and we have to also select the exponents. So this is the equation that we want to model. And for here, we have to put some exponent. Now, here, if you see that for voltage clamps, you are constantly getting increase in the channel currents, but you don't see a decrease. So essentially, there is no inactivation. So we can just remove this equation, part of the equation, so we can have it without an inactivation. So once you make this 0, this part becomes 1. So you only have the activation part here. So we have now started to build the ion channel model. Once you can see now that it has generated some input configuration file, and after a while you will see that it will generate a model file and some responses that it has generated by fitting some parameters to those equations, and those can be then viewed later on. So this is the model, and you can see it has tried to fit the activation steady state activation parameter, and you can see the responses of the voltage clamp stimuli here. So the current look very close to what we have. Now we have built the ion channel model. What would be our next step? Our next step is to build an E model, and then we have to make it simulatable. So for that, let's go and see here this slide. So we have this, this explanation will tell you how it is done. So in this, we can see that this particular process takes almost 4 to 5 hours per run. It's very compute intensive and very time intensive. It uses electrophysiological feature extraction, multi-objective optimization, and some analysis and validation. And since it takes a long time, I've already built it earlier and registered in the webinar project. So what I can do is I can just go to my project. Since I have registered that model here, you will see it under data. It's under project because it's a private data. I'll go under models, and you can see here you have the E model. And now let's confirm if this model has the ion channel that we just built. So we have this description and some metadata, and you can see that we have the exemplar morphology, we have some traces that were used to build the model, and you can see here that this is the KV 7.2 channel. And you also have the SKE2, which is the calcium activated potassium channel, small conductance, and we can use these two channel parameter changes later on to simulate our control and mutation conditions. So the next step was to build an ME model to make it simulatable. So right now, this E model is not simulatable. By the way, you can also check the analysis tab. It has done a lot of validation and analysis to generate this E model. You can see this current scape plots. You can see the optimization that was done to get this model. So you'll understand that it's a very difficult task to make it work. So next step would be to make this model simulatable. So what we do is we go to build, we go to single neuron, and we build an ME model. So for that, what I'll do is I'll just give it a name. And I will go here, M model. And since I have used a morphology which I registered specifically for this E model, the morphology will appear under this project. And you can see here, this is the human neuron which was used. And remember in the beginning, I mentioned that we had human iPSC derived pyramidal neurons that were used. So we have selected a human morphology, and then we will select the E model. We have it here. And then once you build it, it will launch a validation process, and it will generate some validations for input resistance calculation, some FI curve, IV curve calculations. So, and it takes time. So I have already made this before in the interest of time, and I'll directly go here and show you that particular ME model. So I'll go to project. I've registered the ME model myself. So the data occurs under project model, ME model. And then you can see here, this is the ME model. And once you go here, you can see that it is made up of that same morphology from human and the webinar. Now that you have this ME model built, you can start simulating the control and mutation conditions. So this is the interesting part. So we'll go to simulate. We'll go to single neuron beta here this time, and we go to project. And we can see here that our ME model is present. We'll select that. We'll use this particular model. And this is the panel which James was talking about earlier. This is the standard workflow that we have for all our other circuit level simulations. So let's give it a name and start writing. Because we want to make first the control simulation, so I'll do control. I'll just name it, for example, 18.3.2026, so that I remember that this is the name I want to use, because I have to use this name later on for analysis. So it shows you the some initialization parameters. So this is the ME model that was selected. The duration is 1000 milliseconds for simulating this ME model. And now we can select a stimulus. So we have options to select all different types of stimulus, but I want to do very specific somatic current clamp injection. So it's just a current injection. And once we have that, we can just do some parameter filling. I'll just give a 600 millisecond current clamp with a delay of 200 millisecond, and I will give an amplitude of 120% of the threshold current. So once, so the threshold current, the current which will be injected will be 120% of the threshold current. So it will spike. Now I will have to do this experiment multiple times if I want to do the same different current injections. So instead, what I can do is I can do parameter scan, which is a very nice feature here. So I'll just add 150% and I'll add few more of them later on, like this, 170, 200%, and 250%. So I will also have to inject a hyperpolarizing current clamp. So because these neurons have to be held at a holding potential. So we will select the hyperpolarization current clamp, and I will make it for the whole duration of the stimulus. So the holding current of the cell is injected for the whole duration. So now this is done. Now I can just add a recording and start recording the soma voltage. And by the way, you can see on the right, the 3D view, you can move and pan, and you can try to see how it looks like. And then since it's a control condition, I can just simulate it as it is. So let's generate the simulation. What it will do is it will generate the configuration files for all the 5 different amplitudes of threshold currents, and you can see the inputs and outputs later on. So let's just launch the simulation. And once you have launched it, it will start running in some time, and it will generate the voltage recordings and some NWB files which contain the voltage recordings, which you can view later on. So now the simulations are running, all 5 simulations simultaneously, and then in some time you will see that 3 different files will be generated in the output files tab, and you can see the voltage result in this NWB file. So you can see these are the traces for 120% current injection. You can try to zoom in and to analyze here. You can do the same thing for other simulations, and you will see the outputs like this. So now we have simulated the control condition. The next would be to simulate the mutation conditions. So what I can do is I can go back to this configuration tab, and I can just do this new simulation campaign. So what it did was it copied the same configuration that I had before and just make it available for me to change it. Now I can just change here some things. So because I want to simulate the mutation conditions, I will just change it to mutation, and I will leave the initialization as it is. The stimuli will also remain as it is. You have hyperpolarization, current injection, you have the recordings. And now here we want to change the conductances. So we have neuronal manipulations. We'll select this one. It gives you option to select two different types of manipulations. Either you can do manipulation by section list, which can be for the whole axonal tree, for the apical tree or basal tree, or you can do it for the full neuron. We'll do this section list type. So here it gives me an option to select different ion channels. Now, these are the ion channels that we saw in the detailed view sometime back. So my channel of interest is this, KV 7.2. And by the way, if you want to know more about it, you can click on this link. So it will open a new tab, which you can then see here, the ion channel details. But yeah, let's go back. We'll come here, we'll select the ion channel. This particular variable is the maximum conductance. So we select that. And since we want to reduce the conductance of that particular channel, what we can do is we can change value to any other value that you want. So these are the default values. And let's just try to make it approximately half. So I'll do that. And I'll also change it for this one. Now, the next manipulation that I want to do is for the SK channel. I'll do the same type of manipulation for SK channel. I go to this and I know that this particular channel is SK channel, SKCA, which is the calcium activated potassium channel that you saw earlier in the experimental study. And it was said that the conductance of these channels increase. Now I can just change it by making it double of the value approximately. I'll make it 0.8 and I will just make it 0.13. Now you have these two manipulations added to the same simulation and I will generate those simulations. So it will do the same thing. It will generate 5 different simulation campaigns. 5 different simulations in the campaign, which you can launch. And then once you have launched, it will start running. Again, it will generate those output files, which you can then view and analyze. So it's now pending. So it's going to run on the cloud. And this will run in parallel. These are now running, all of them. And in a few seconds, they will be done and you will see 3 different files which will have the spikes, the soma recordings that you earlier recorded, and you can visualize the traces recorded in the NWE files. So you can already see that there might be some difference in the spikes, but you are not really sure. So what we can do, this is the next step. So we can go to notebooks and analyze those results. So we go here. I've specifically registered a notebook for this particular webinar. I click on this plus, and then I'll click on run. So what it does is it launches a Jupyter Hub server with that notebook open. All the packages are pre-installed, and you can just run that notebook along with the simulations that you have just done, and you can get the results. So start running it. The first one tells you what virtual lab and project ID you are in. You start running that. There are some imports. Now, since the data is private to you, you have to authenticate yourself so that you give access to that notebook. So you have now this authenticated, and now I have to select the project in which I did the simulation. So it is this project which I ran in. So now you have to select those campaigns that you just ran. You can query this from our database by just running this, and you can already see the final campaign that was run some time back for control and for mutation. So we ran that, we select that, And now we want to download those campaigns locally so that we can analyze them. So it will download the control and mutation simulation campaigns. You will have the output recordings in it, which you can then analyze further. You can already see that there is this folder created, and this particular cell actually is doing a query from our database to find this particular simulation campaign with this name, mutation and control. And you can see it's as already finished. Now let's do a side-by-side comparison. So you can see here for this 120% simulations, there are some differences. 150%, the number of spikes looks slightly different. And now you can do some more further analysis by trying to compare single APs. So this is the control and mutation simulation for one AP comparison. And this is the result from the experimental paper. You can see there are some differences, and that difference could be because we have not simulated the BK channel, and BK channel is known to take part in this initial phase of the AHP. So our goal was mainly to see whether we can try to simulate these conditions on this platform, and we have almost made it possible to regenerate the same result that we have in the experiments. You can do further analysis later on. So we can invite you to this project, and you can also do some further analysis by extracting some features, and you can also plot them later on. So you can see here, these are the features extracted for different simulation amplitudes, and these can be then analyzed further. So this tells you how you can do simulations for mutation and control campaign. And similarly, you can do other pathological condition studies using the Open Brain Institute virtual labs. So I will hand over to Oliver, and I'll go back to the slides here so that we can directly, so the question and answers will be at the end. If you have any questions, do post them in the chat. And these are the references. Now, Oliver can take over.
Thank you, Darshan. It was really interesting to see you use the platform as well, so thank you for that. But hi, everyone. It's nice to meet you, and I hope you're all well. My name's Oliver. I'm here with my colleague Sara as well to speak to you a little bit more about Frontiers. I'm the journal specialist working for Frontiers in Computational Neuroscience. And for those less familiar with the journal focuses on advancing our understanding of brain function through computational approaches, and that includes everything from biologically realistic simulations to AI-driven methods for understanding mental health conditions. The journal's led by chief editors Si Wu and Misha Sodykes, and we're also supported by a global editorial board full of experts across computational neuroscience. In terms of what we can offer you as a journal, there are several ways to get involved. So first of all, we always welcome article submissions across a range of article types. So if you have an article that's in preparation or ready to be submitted, feel free to reach out to us using the email address at the bottom right here. And we'd be happy to help you find a suitable research topic for your work. Publishing within a research topic really helps increase the visibility and impact of your research. And on top of this, for some of our more established or high impact research topics, we can also offer you a discount on your submission as well. So do reach out to us if you have any work that's on the way. But beyond submitting your own work, you can also have the opportunity to collaborate with us in a further editorial role. This can take different forms depending on the level of involvement that you're wanting. But for those interested in a more long-term collaboration, we can invite you to join our editorial board, and this is a great way to contribute directly to the journal, open access science, supporting the peer review process of your colleagues and things like this. But if you prefer something more focused or kind of a little bit more of a smaller project to begin with, you can also get involved by editing a research topic as well. So whether you'd like to contribute as an author or take a more temporary editing role for a research topic or join the editorial board as an editorial board member, we'd be very happy to explore the best way to go about that together. And finally, we're also very proud to partner with the Organization for Computational Neurosciences, and we're continuing to develop opportunities within the community through initiatives such as society-led research topics. So if you're interested in hosting a research topic in collaboration with the Organization for Computational Neurosciences, please use the link that is on our slide here, and you can register your interest in doing so there. And if you're attending as well, feel free to let us know, because we'd be really happy to meet you and speak about your work in person as well. Thank you all very much, and I'll pass it over to Sara.
Okay, next slide, please. Hi, everyone, and nice to meet you. My name is Sara. I'm Journal Manager for Frontiers in Neuroinformatics, Molecular Neuroscience, and Neuroergonomics. And today I'm here to represent Frontiers in Neuroinformatics, which is a completely open access journal, one of the first launched by Frontiers back in 2007, and led by our field chief editors, Jan Bjali from the University of Oslo, Sean Hill, co-founder of Sense Science, and Michael Denker from Jülich Research Center. Probably, since it's 2026, you have your phone close to you. So if you want, you can scan the QR code on the right side of this slide to access the journal page directly. You will see our many articles published, more than 1,000, and also our currently open projects. The journal has an impact factor of 2.5 and a site score of 5.5. And in terms of scope, we focus on applications and technologies that facilitate data sharing, developmental data and knowledge bases for all levels of neuroscience, neuroscience databases, both basic and clinical, informatics approaches that merge clinical and basic neuroscience data, and novel tools for data acquisition, analysis, visualization, and dissemination of nervous system data. We accept many article types, as you can see listed here. They are divided into two main categories. Type A include original research, systematic reviews, methods, reviews, hypothesis and theory, technology and code. And type B include mini reviews, perspective, data report, brief research reports, general commentary, and opinion pieces. We also now accept Fair Square articles. This is the new service provided by Frontiers that combines both a data management service and a publishing service. And we look forward to publish the first Fair Square article in the journal. So if you're interested in being the first authors to do this, please feel free to reach out. As Oliver said, we are also very open to discuss collaboration, such as associate editor roles, reviewer roles, depending on your seniority, and also research topic collaboration. You can collaborate both as a topic editor, proposing a topic, but also participating as author. We currently have 6 research topics open. Research topics are article collection, different from the standard special issues, for giving more flexibility to both authors and editors, and for accepting all the article types that you see here. The topics that we currently have open span from fair neuroinformatics, data management, AI, EEG techniques, but we also have others currently under development. So, and as Oliver said, we also have the possibility sometimes to provide discount for confirmed contributors. And on top of that, we can always provide personalized deadline extension. So if you're interested in discussing any of these points, feel free to reach out. You can contact us at neuroinformatics@frontiersin.org, or you can reach out also to me directly. My email is sara.donofrio, so name.surname @frontiersin.org, or feel free to reach out and connect on LinkedIn. Always happy to connect. Thank you everyone for your attention, and especially thanks for the Open Brain Institute for having us here in this very interesting webinar.
Thank you both. Yeah, very nice. Much appreciated. So firstly, before we go to the Q&A, we'll just point out that, through this QR code, you can head to the virtual labs, create your own virtual lab. There's links to our LinkedIn and YouTube, where you can see more content and see our updates. Particularly, we have a follow-up webinar coming in around a month's time from our Chief Scientific Officer, Michael Ryman, about spines and morphologies. With that, we can start answer some of the questions which we've published. So the first question we had was from Carolina about the computational cost of running single neuron simulations. I think Darshan mentioned the optimization of single neuron models. So that optimization process is expensive, but usually in these types of studies, you optimize one or a small group of neurons, and then you generalize those electrical models to other neurons. So really, you only have to perform that process for a small subset of neurons, and then you apply the electrical models to other neurons because of the similarity between morphologies, and you then validate that they within the biological expectations. And the key point is that our platform is full of these neurons which have already been optimized, so you can go straight to simulating them, and simulating them for a virtual experiment is computationally very, very cheap.
Yes.
So with the free 100 credits you get when you sign up, you can simulate about 100, make about 100 single neuron simulations already. So then, I also saw we had a question from Katerina Juka, who we've discussed the platform several times before. So nice to hear from you, and thank you for joining. So the analyses we saw within the platform, they are kind of state-of-the-art analyses defined by us. But the hope is that we eventually allow users to run automated analyses and propose automated analyses for the community, which appear on the platform. But already you can run your own analyses on notebooks in the platform, and very soon you'll actually see the results of those notebooks appearing in the platform as well. So Darshan, you may want to answer.
Yeah. Okay, so there is one question by Gabrielle. So it's if I was wondering if it would be easy, as explained, to emulate mutation inducing a gain of function by shifting the activation function of the channel. Yeah, so actually you can do that. So as I showed you before, maybe I can quickly just go here and show that. So in the workflow, you have options to select different types of variables. So if I go to my model and use that model, and once you have that model as a range variable, so we are using the neuron simulator here, and range or global variable, they will appear here automatically. So if you have a variable for shifting the activation or inactivation parameters, you can see them here, and you can change it from the default value to the different value that you want to test. So I guess that is possible. Do let me know if you have further comments, or if you want to discuss it further, you can also reach out to me. Let's take some other questions.
This one about.
So for the Jupyter notebooks, so this is from Carolina again. For Jupyter notebooks with the analysis, can you have it compare the simulations of two different cell types, or does it only analyze one simulation at a time? So as you saw here, you can actually do that. You can compare two different cell simulations. You can even do two-circuit simulation comparisons, or you can do multiple simulation comparisons across neurons or across circuits within one single notebook by getting the simulation campaigns of all those neurons and then analyzing it together. So it is possible. So right now I used only one neuron here, but you can do another neuron simulation and you can just try to filter it here, and then you can do get it.
Yeah, so here we can see where we are fetching the two different simulations. So yeah, you can select multiple simulations and compare them in this way.
Yeah. Yeah,
Yes.
You can change the query here and get different simulation based on the name or the type of the model that you have used, the name of the model. So it's pretty easy to get different neuron simulations.
Yeah, and you can copy the ID as I showed in the top right after the simulation and use that to load the.
Yeah, let's show that. So yeah, I have to. I have to go to workflows table and I can go here. First I go simulate and go single neuron beta. So this shows us the recently run simulations. So you can see here, you can directly view the currently the simulation that you just ran. And here is the simulation and you can copy the ID from here. So you can directly take that ID, create a query and get the simulation here. This is what this particular step is doing. So yeah, you can do a query for different neurons. So we'll move to the next question.
We had another question from Carolina about simulating transcranial stimuli. So currently that's not supported in the platform, but modeling of stimuli with finite element models of the skull and scalp we have done before and hope to at the Blue Brain project and hope to reintroduce into the platform. But already we have extracellular electric fields you'll be able to apply to the larger simulations very soon, and we've already implemented that, and also to eventually the smaller simulations. But you can also kind of for now model that as a proxy of just current injections into somas perhaps. But if that's a particular use case you have in mind, definitely reach out to us and we can see how we can support that. So looking at the other ones, got tick. Which ones have we missed? The top one.
The new post. Can you click on this?
Oh yeah, new posts. Okay. Posters, ion channel.
It's possible to use more complex customized ion channel kinetics models for specific conductances. Yes, so I think you have in mind different type of equations that can go into the model. So maybe let's show one of the ion channel models. So I'll show you the ion channel model that we just created. I'll go to project, go to model, go to ion channels, and you can see here, this is the model you just built. And you will have the same figures that you saw when you generated them. And this is the file. So I guess what you meant by more complex and customized ion channel kinetics is by changing these parameters, the equation types. So yes, we can support that. We can add different equations to the workflow during the building phase. So we have, like right now, these 4 different types of equations for different types of parameters. We can add more if you want more, or if you have any particular different type of modeling, like the Markov modeling of ion channels. We can also think of supporting that. If you have any particular use case, do reach out to us back and we can let you know.
And you can also upload your own models to the platform. directly using Entity SDK, our API for interacting with the cloud database. So there's another question from Raphael about, thank you, really cool platform, appreciated. So enterprise offerings, so use your own cloud resources. So we do plan to put this onto the marketplaces for Azure and AWS, which should enable this, but that's not here yet. Yeah. Okay.
Okay, one more.
One more. Fabrice. So yes, that is actively being worked on to allow LFP recordings in a microcircuit simulation. So these virtual extracellular electrodes I spoke about, like the NeuroPixels electrode, which is in press, our paper in press. We're actively working on adding that to the platform, and that would support recording LFP as well. How long would it take for a 1 second simulation from a circuit that includes 1,000 neurons, 800 interneurons? So that compared to the simulation at Blue Brain, which had 210,000 neurons, that's relatively light. So I'm afraid I don't have an exact estimate, but it would be relatively quick. Yeah.
Real time.
Yeah, not quite real time, but you'd probably for each second of biological time, maybe on the order of a minute.
10 to 1.
10 to 1. We have a quote in the room. 10 to 1. Yeah. Okay, one more. So is it possible to simulate long-term plasticity? So yeah, we've had a lot of people asking for this feature. So yeah, that was described in one of the papers I spoke about. That's not supported yet on the platform, but we're actively working on adding it. Yeah. Okay, is that everything? So if you have features which you would like us to support to help you accomplish your scientific objectives, definitely let us know. Any feedback you have is really useful. So yeah, please be in touch, leave feedback, and if anyone else has a question, maybe you can raise your hand now. If not, we can leave this first webinar there. Yeah, and we have in around a month's time, our Chief Scientific Officer talking about spine extraction and analysis of spines on neurons, which is really new and very exciting work. So yeah, if you sign up to the platform, if you register to our newsletter or just sign up, you'll receive notifications and you'll certainly see it. Great. So thank you very much for joining. Thank you to Sara and Olivier for their presentations. And yeah, we hope you enjoy your first virtual experiments and hope to see you at the next webinar in around a month's time. Thank you.
Thank you.